In this second podcast in our series on the female reproductive system, we're going to talk about the ovarian follicles. Now, as you know, the ovarian follicles provide the microenvironment for developing oocytes. We talk about primordial follicles that contain oocytes that are arrested in diplotin of meiosis I. There is one oocyte per follicle. There are approximately 5 million follicles that form by the 4th to 5th month of fetal life. But oocytes and ovarian follicles are essentially a decaying cell population. By birth, a baby girl has 600,000 to perhaps 800,000 follicles. Follicular atresia begins around the 5th month of fetal life. A woman ovulates perhaps 400 mature ova during her reproductive life, and ovulation usually begins about 1 year after the onset of menarche. Follicular development has 3 basic stages that we're going to review. There are primordial follicles, there are growing follicles, considered early and late primary follicles, and then secondary or antral follicles. We'll talk about those. And then there's the mature or Graafian follicle stage. This cartoon just shows early follicular development showing primordial follicles, and here histologically would be some of the primordial follicles. An early primary follicle, here showing an early primary follicle, and then a late primary follicle shown on the bottom histological image. Now, in general, the primordial follicles' growth is independent of gonadotropin stimulation. There is a single layer of squamous follicle cells that surround the oocyte. You can see that in the histological view like this, the single layer of squamous follicle cells. Primordial follicles are located in the stroma just beneath the tunica albuginea. There are early primary follicles which have a single layer of cuboidal follicular cells. Note the single layer of cuboidal cells around these early primary follicles, and you begin to see the formation of a zona pellucida. The zona pellucida is composed of 3 specific glycoproteins. We'll talk about these a little bit later, but there's ZP1, ZP2, and ZP3. And then late primary follicles, the follicular cells undergo rapid mitosis and they stratify to form a layer of granulosa cells. And you can see this layer of granulosa cells around this later stage, late primary follicle. You can clearly see the zona pellucida like so. Now, an interesting point, these granulosa cells do not have zonular occludens between them. They have a lot of gap junctions between them, but there is no zonular occludens between the granulosa cells, so there's no blood follicle barrier. This is different than in the male system where there was a blood testis barrier that allowed the developing spermatocytes to be in an immunologically privileged location. That is not the case with the developing oocytes. There is no blood follicle barrier. In these primary follicles, you can see here, this one, you can see the oocyte. You can see this one has a profile of the nucleus in the plane of section. This younger one also has a profile of the nucleus in the plane of section. You don't always see nuclei in the plane of sections when you're looking at oocytes because oocytes are fairly large cells. Here's a primordial follicle, both diagrammatically and histologically. The primordial follicles appear during the 3rd month of fetal life. As we said, the oocytes are arrested in diplotin I of meiosis. Very characteristic of primordial follicles is an eccentrically placed nucleus. You can see that in the diagram, but you can see that nicely in this histological view. This one is eccentrically placed. This one is clearly eccentrically placed, as is that one. Notice the squamous follicular cells that surround these young primordial follicles, drawn diagrammatically like this. The follicular cells are separated from the surrounding connective tissue by a basal lamina, and that's shown in the diagram like that. Here, the oocyte might have a size of about 30 microns in diameter, so 30 microns diameter like that. so these would be approximately 30 microns in diameter. There's a Balbiani body, which is very characteristic of these young primordial follicles. The Balbiani body consists of the ER, Golgi, and multiple vesicles. And then there's also something that appears to be what are called annulate lamellae. You don't see these histologically, but on good high-power electron micrographs, you see these annulate lamellae. They appear to be stacked pieces of the nuclear envelope. They're like nuclear envelope-like membranes that actually have pore complexes associated with them. We don't really know what the function of that is. We do know that as the oocyte is getting larger, there is more of a demand for membranes, but to my knowledge, no one has ever really described the function of the annulate lamellae in developing oocytes. And of course, these young primordial follicles do have numerous small vesicles and mitochondria. Here's an early primary follicle, both histologically and in the diagram. Here the oocyte is enlarging. It is now perhaps 50 to 80 microns in diameter. The follicular cells are beginning to proliferate and they're becoming cuboidal. That's clearly visible in the histological image, and it's also nicely visible on the cartoon. The oocytes are now actively transcribing ribosomal RNAs. I hope you'll remember that early embryos are developing primarily only with the maternal ribosomes, and those ribosomal RNAs are being transcribed early in oogenesis. The basal lamina of the follicle cells continues to maintain the boundary between the follicle and the surrounding connective tissue, and you can see this basal lamina nicely here on the histological image. The basal lamina tends to stain PAS positive. The zona pellucida is clearly beginning to form on the early primary follicles. You can see little profiles of the zona pellucida here. And the zona pellucida contains spermatozoan binding proteins, and they induce the acrosome reaction. So it is rather interesting that the zona pellucida, with these important proteins for spermatozoa binding and formation of the acrosome reaction, are forming really very early in the development of the follicles, sometimes almost decades before they're going to come into play as a potential ovum is being fertilized. Here's a later stage primary follicle. As the granulosa cells are proliferating in these later stage primary follicles, the stromal cells are going to begin to form a connective tissue sheath, which is going to be called the theca folliculi, and that's just external to the granulosa cell basal lamina. So here would be the basal lamina in the diagram of the granulosa cells, and this would be the theca folliculi. The theca folliculi is eventually going to form the theca externa and the theca interna. As we'll come to see, the theca interna is a highly vascularized layer. The cells in the theca interna have characteristics of steroid-secreting cells, and they have receptors for LH. The cells of the theca externa are essentially connective tissue cells, smooth muscle cells, and there are bundles of collagen fibers in the theca externa. The oocyte at the late primary follicle stage is metabolically very active. There are numerous Golgi, there are abundant ribosomes, and this is when you begin to see the formation of the cortical granules. Here in the histological image you can see a profile of the nucleus in the plane of section. You can see nucleoli, and you can clearly see the chromatin spun out into this relaxed diplotene configuration, again suggesting that there's active transcription of ribosomal RNA off of these diplotene chromosomes, at least on the ones that are coding for the nucleolar organizer regions. Microvilli of the oocyte and granulosa cells intermingle with each other in an area called the Perivitelline space, but there's no cytoplasmic continuity between the oocyte and the granulosa cells. And that's kind of shown here on the diagram. So this would be processes of the granulosa cells like that, and here would be some microvilli from the oocyte membrane. This cartoon is just like a higher power image, if you want, of the perivitelline space to show the interactions of the follicular cells, so-called granulosa cells, with the oocyte membrane, the so-called oolemma. And you can see this perivitelline space around the oocyte and between the follicular cells. Here are follicles in a later stage of development. So here's what's called a secondary or antral follicle and a mature or Graafian follicle. Characteristics, you can see the histological images of the secondary or antral follicle. You're beginning to see a space that's forming, and we'll talk about this in just a second, but you're beginning to see this antral space forming in these later stages of follicular development. When you see the antral space, we call this a secondary follicle. Now be very careful here. We talk about primordial follicles, primary follicles, secondary follicles. These terms are unfortunate terms. They don't relate to stages of meiosis. Everything that you see within the ovary would still be considered primary oocytes arrested in the very first meiotic division in diploatine. The mature or Graafian follicle, this antral space is getting very much more expanded, and that's evident in this histological image. You're beginning to see something called a cumulus ophorus. We'll talk about that in a few minutes, and a corona radiata. We'll also talk about that in a few minutes. A little more detailed view of the secondary follicle. As we said, the antral space is forming. The secondary follicle is moving deeper into the cortex. This antral space is becoming filled with a fluid, a so-called liquor folliculi. This liquor folliculi is rich in hyaluronic acid. Now, the oocyte and follicle cell growth depends on FSH and various growth factors and calcium. Some of the growth factors that are involved are epidermal growth factor and insulin-like growth factor. There can be up to 12 layers of granulosa cells that you'll see in the follicle. The follicles are going to grow from about 0.2 millimeters in diameter up to 10 millimeters in diameter. So they're getting quite a bit larger. Oocyte maturation inhibiting proteins are also secreted by the granulosa cells into the antral spaces, and that's going to keep follicular growth in check. And this is interesting because there are, as we said, at birth something like 400 to 800,000 oocytes in follicles, but they're not all growing at the same time, and it turns out that there's higher concentrations of this oocyte maturation inhibiting protein in the younger follicles. We don't really know what causes the follicles to mature, so we don't know what makes some of the follicles have lower concentration of this oocyte maturation inhibiting protein. These secondary follicles, as we said, might be up to 10 millimeters in diameter, but the oocyte diameter is only about 125 microns. Not so evident in the histological view, but here is the theca interna and the theca externa. The theca interna is vascularized. It's a little bit hard to see it in this histological view, but the cartoon is showing that, the blood vessels within the theca interna. On the cartoon, you can see the basal lamina, so this would be the granulosa cells, so the theca interna, the theca externa. And then here's something I'd like to call your attention to. They're called Karl-Exner bodies. These Karl-Exner bodies are extracellular conglomerates of junk. It's just extracellular material. But they are seen in abundance in tumors that originate from granulosa cells. So ovarian tumors that have a granulosa cell origin have large numbers of these Karl-Exner bodies. You don't have to remember that for our course, but it may be something you want to file away. You may see that on licensing exams down the road. Here's an example of a mature or Graafian follicle histologically. Look at the large antral space and look at the T9C oocyte in relation to this large follicle. The follicle diameter is more than 10 millimeters, and it would be bulging from the surface of the ovary. Luteinizing hormone stimulates theca interna cells to secrete androgens. This leads to the secretion of FSH from the anterior pituitary, and the granulosa cells will convert androgens into estrogens, and that's going to stimulate granulosa cell division, so the follicle is going to get larger and you'll get greater numbers of these granulosa cells. the LH surge from the anterior pituitary is going to trigger the first meiotic division, so you'll actually form the secondary oocyte, and ovulation is going to occur either at that time or just a few hours later. So I told you a few minutes ago that all of the oocytes that you see within the ovary, within follicles, would be arrested in the first meiotic division. There'd be primary oocytes. This would be an exception to that these large Graafian follicles, they could potentially be secondary oocytes, and the timing of this is not fully clear. The LH surge does trigger the first meiotic division. In many cases, the first meiotic division is occurring simultaneously as the oocyte is ovulated. Note here in the histological view and in the cartoon, the granulosa cells are thinning out around the periphery of the follicle, but they're mounding up in a large pillar, if you want, under the follicle, the so-called cumulus ophorus. That's just a mound of granulosa cells on which the oocyte rests, and it rests on that cumulus ophorus surrounded by a couple of layers of granulosa cells, the so-called corona radiata. Now, during ovulation, the oocyte and these cells of the corona radiata are what are released from the ovary.